Hello everyone, and welcome to today's session. I'm excited to have you here as we explore the fascinating world of the chemistry of nutrients. Today, we'll be focusing on Module 2, specifically the chemistry of nutrients. In Study Session 4, we'll dive into the chemistry of amino acids and proteins, which are fundamental to understanding biochemistry. This is part of the BCHM 241 course on the biochemistry of macromolecules. Over the next few minutes, we'll cover the key aspects of amino acids and proteins, their structure, function, and importance. So, let's get started and discover the essential chemistry behind these vital molecules. Let's start by exploring the chemical nature of amino acids. These molecules are the building blocks of proteins, and there are 20 different amino acids commonly found in proteins. Each one has a unique side chain, or R group, which gives it distinct chemical properties. Understanding these properties is crucial because they influence how amino acids interact and fold into functional proteins. Next, we'll look at the chemical properties of amino acids themselves. These properties include aspects like polarity, charge, and hydrophobicity, all of which affect protein structure and function. For example, some amino acids are polar and can form hydrogen bonds, while others are nonpolar and tend to be buried inside the protein. Now, let's discuss the pI, or isoelectric point, of amino acids. The pI is the pH at which an amino acid carries no net electric charge. This is important because it influences solubility and how amino acids behave in different pH environments, especially during protein purification or analysis. Moving on, we come to the formation of peptide bonds. These are the covalent bonds that link amino acids together in a chain. The formation occurs through a dehydration reaction, where a molecule of water is removed. This process is fundamental because it creates the primary structure of proteins, dictating their eventual shape and function. Throughout this presentation, understanding these concepts helps us grasp how amino acids serve as the foundation for all proteins, influencing their structure, stability, and activity in biological systems. Amino acids are a fascinating group of organic compounds that contain two key functional groups, the amino group and the carboxyl group. These building blocks are essential because they form the basic structure of proteins, which are vital for countless biological functions. Proteins can be broken down, or hydrolyzed, into their individual amino acids using various methods, which is really important for understanding how our bodies digest and utilize proteins. It's also interesting to note that some amino acids in proteins are modified forms, which can influence how proteins behave and their functions. In nature, we typically find 20 different amino acids in proteins, each with unique properties that contribute to the protein's overall structure and function. Now, these amino acids don't just float around independently. They are linked together by peptide bonds, forming chains called polypeptides. When amino acids are connected this way, the sequence of amino acids is crucial because it determines how the chain will fold. This folding leads to the secondary and tertiary structures of proteins, which are essential for their specific roles in the body. So, understanding amino acids and their connections helps us grasp how proteins are built and how their structure relates to their function. This foundational knowledge is key to appreciating the complexity and elegance of biological systems. Let's take a closer look at the chemical nature of amino acids. All amino acids share a common structure. They have a carboxyl group and an amino group both bonded to the same carbon atom, which we call the alpha carbon. What makes each amino acid unique are their side chains, also known as R groups. These R groups can vary quite a bit. They differ in their structure, size, and electric charge. This variation is really important because it influences how soluble the amino acids are in water, which in turn affects how proteins fold and function. There are 20 amino acids that are found in proteins, and each of them has a specific 3-letter abbreviation and a 1-letter symbol. This helps us identify them quickly in sequences. Now, when we look at the R groups, any additional carbons attached are often labeled with Greek letters like beta, gamma, delta, epsilon, and so on, indicating their position relative to the alpha carbon. This systematic naming helps us understand the structure and properties of each amino acid better. Understanding these structural differences is fundamental because they influence how amino acids behave in different environments and how they contribute to the overall structure and function of proteins. This is a key piece of the puzzle in understanding biochemistry and molecular biology as a whole. Let's take a closer look at the structure of amino acids. For all the common amino acids, except glycine, the alpha carbon is bonded to 4 different groups. These include a carboxyl group, an amino group, an R group which varies among amino acids, and a hydrogen atom. Because of this, the alpha carbon is what we call a chiral center, meaning it has a specific 3-dimensional arrangement that makes it asymmetric. Now, this tetrahedral arrangement around the alpha carbon allows the 4 different groups to occupy 2 distinct spatial arrangements. Essentially, this means amino acids can have 2 different stereoisomers, which are mirror images of each other. In stereochemistry, for all chiral compounds, the stereoisomers are classified as either L or D. This designation relates to the configuration of the molecule compared to glyceraldehyde, a simple sugar. If the stereoisomer's configuration is similar to that of L-glyceraldehyde, we call it an L-amino acid. Conversely, if it resembles D-glyceraldehyde, it's a D-amino acid. Understanding these stereoisomers is crucial because they can influence how amino acids behave in biological systems, and, as part of the larger picture, they play a significant role in protein structure and function. Today, we're looking at the fundamental building blocks of proteins, the 20 amino acids. These are the basic units that combine in various ways to form the diverse proteins found in all living organisms. Each amino acid has unique properties that influence how proteins fold and function. Understanding these amino acids helps us grasp how proteins achieve their specific shapes and roles. Remember, the sequence and type of amino acids determine a protein's structure and activity, which is central to the overall theme of how biological molecules work together to sustain life. Let's take a closer look at the structure of amino acids. First, we have amino acids with aliphatic side chains, which are non-aromatic and typically flexible, contributing to the overall shape of proteins. Next, amino acids containing hydroxyl groups are important because they can participate in phosphorylation and other reactions. Sulfur-containing amino acids, like cysteine and methionine, play crucial roles in protein structure and enzyme function. Then there are acid amino acids, such as glutamic acid and aspartic acid, along with their amides, which are involved in neurotransmission and metabolism. Moving on, we have basic amino acids like lysine, arginine, and histidine, which are positively charged at physiological pH and often participate in enzyme active sites. Aromatic amino acids, including phenylalanine, tyrosine, and tryptophan, are notable for their ring structures and their roles in light absorption and signaling. Amino acids, such as proline, have a ring structure that influences protein folding and stability. Now, let's consider the polarity of amino acids. Some amino acids are polar, meaning they can form hydrogen bonds and are often found on the exterior of proteins interacting with water. Others are nonpolar, residing mainly in the interior of proteins to maintain hydrophobic core stability. Next, we look at the nutritional requirements. Some amino acids are classified as essential because our bodies cannot synthesize them, so they must be obtained from our diet. Non-essential amino acids can be produced internally and are less critical to obtain directly from food. Finally, let's discuss the metabolic fate of amino acids. Some are glucogenic, meaning they can be converted into glucose during gluconeogenesis, while others are ketogenic, which are converted into ketone bodies. There are also amino acids that are both glucogenic and ketogenic, providing versatility in energy production. All of these classifications, structure, polarity, nutritional importance, and metabolic pathways, help us understand how amino acids function in our bodies and their critical roles in health and disease. Let's take a closer look at amino acids with aliphatic side chains. These are amino acids that have non-aromatic, straight or branched hydrocarbon chains in their side groups. They are generally nonpolar and hydrophobic, which means they tend to avoid water and prefer to be buried inside the protein structure. This characteristic is crucial because it influences how proteins fold and maintain their stability. Notable examples include glycine, alanine, valine, leucine, and isoleucine. Each of these plays a specific role in protein architecture, often contributing to the hydrophobic core that drives proper folding. Recognizing these amino acids helps us understand how proteins achieve their functional three-dimensional structures, which is essential for their biological activity. So, by studying amino acids with aliphatic side chains, we gain insight into the fundamental principles of protein chemistry and stability. Let's take a closer look at amino acids containing a hydroxyl group. These amino acids are quite interesting because the hydroxyl group, which is a chemical group consisting of oxygen and hydrogen, adds unique properties to these molecules. The hydroxyl group makes these amino acids more polar, which can influence how they interact within proteins and with other molecules. This polarity often affects their solubility and their role in enzymatic reactions. For example, serine and threonine are two amino acids that have hydroxyl groups. These amino acids are not only building blocks of proteins, but also play vital roles in cell signaling and enzyme activity. Understanding these amino acids helps us grasp how proteins can be modified or how they participate in complex biological processes. So, recognizing the presence of hydroxyl groups gives us insight into their functional versatility and importance in biochemistry. Let's take a closer look at sulfur-containing amino acids. These amino acids are unique because they include sulfur atoms in their structures, which play important roles in the body. The two main sulfur amino acids we focus on are methionine and cysteine. Methionine is essential, meaning our bodies can't produce it, so we need to get it from our diet. It serves as a building block for other sulfur-containing compounds and is crucial for methylation processes. Cysteine, on the other hand, is considered semi-essential because our bodies can produce it from methionine, but it's also obtained through diet. Cysteine is vital for forming disulfide bonds that maintain protein structure and stability. These amino acids are not only important in protein synthesis, but also in antioxidant defenses, thanks to compounds like glutathione. Overall, sulfur amino acids are fundamental for maintaining cellular health, supporting structural integrity, and facilitating metabolic processes. They truly highlight the interconnectedness of amino acids and overall health in our presentation. Let's take a closer look at acid amino acids and their amides. These are a special group of amino acids that have unique properties due to their acidic nature. Acid amino acids, like glutamic acid and aspartic acid, contain carboxyl groups that can donate protons, making them important in metabolic processes and enzyme function. Now, their amides are formed when the carboxyl groups are converted into amides, resulting in compounds like glutamine and asparagine. These amides play crucial roles in nitrogen transport and metabolism within the body. Understanding these structures helps us see how amino acids contribute not just to protein synthesis, but also to various biochemical pathways. So, recognizing the differences between acid amino acids and their amides enriches our overall understanding of amino acid chemistry and their biological significance. Now, let's take a closer look at the basic amino acids. These are the building blocks of proteins that are essential for many biological functions. There are 9 standard amino acids, but the focus here is on the core group known as the basic amino acids. These are characterized by having side chains that are positively charged at physiological pH, which makes them particularly important in processes like enzyme activity, signal transduction, and maintaining protein structure. The key players in this group are lysine, arginine, and histidine. Each of these has unique properties that allow them to interact with negatively charged molecules, like DNA or other proteins, which is crucial for cellular function. Recognizing these basic amino acids helps us understand how proteins fold, how they interact with other molecules, and their overall role in cell biology. This understanding ties into the broader theme of how amino acids influence the structure and function of proteins in living organisms. Today, I want to talk about aromatic amino acids, which are a special group of amino acids characterized by their aromatic rings. These include phenylalanine, tyrosine, and tryptophan. What makes them particularly interesting is their unique structure, which contains these ring systems that can absorb UV light efficiently. This property is often used in laboratory analysis to track and quantify these amino acids. Aromatic amino acids play crucial roles in protein structure and function because their rings can participate in stacking interactions, stabilizing the three-dimensional shape of proteins. For example, tyrosine and tryptophan are not only structural components, but also involved in enzymatic activity and signaling due to their ability to participate in electron transfer and hydrogen bonding. Connecting this to the larger picture, understanding these amino acids helps us grasp how proteins fold and function at a molecular level. Their aromatic nature contributes significantly to the stability and reactivity of proteins. So, in essence, aromatic amino acids are more than just building blocks. They are key players in the complex chemistry of life processes. Let's talk about amino acids. These are a special class of amino acids that play unique roles in protein structure and function. Unlike the typical amino acids with amino groups, amino acids contain a nitrogen atom in a different position, which influences how they behave. One of the most well-known amino acids is proline, which is crucial in stabilizing the structure of proteins like collagen. Because of its rigid ring structure, proline introduces kinks or bends in protein chains, affecting how proteins fold and maintain their shape. This is really important when we consider how proteins achieve their functional three-dimensional forms. Overall, understanding amino acids helps us grasp some of the more intricate aspects of protein chemistry and how their structures are maintained and regulated. Let's explore some key properties of amino acids that are essential for understanding their roles in biochemistry. First, solubility. Most amino acids are soluble in water because they can ionize in solution, which means they can carry a charge and dissolve easily. This is important because it affects how amino acids behave in biological systems. Next, taste. Some amino acids have distinct tastes. For example, glycine and alanine are sweet, while others like leucine or phenylalanine can taste bitter. Interestingly, some amino acids are tasteless, which also plays a role in how proteins and peptides are perceived. Moving on to light absorption, certain amino acids such as phenylalanine, tyrosine, and tryptophan can absorb light at 280 nanometers. This property is useful in spectroscopic studies, helping us quantify and analyze proteins in a solution. Now, let's talk about the formation of esters. One example is monosodium glutamate, which is a sodium salt of glutamic acid. It's used as a flavor enhancer and illustrates how amino acids can be chemically modified for practical applications. Decarboxylation is another interesting process. For instance, amino acids like histamine and tyramine are produced through decarboxylation, which is removal of a carboxyl group. These compounds can have significant biological effects, such as acting as neurotransmitters or influencing blood pressure. Then we have transamination. This is a key process where amino groups are transferred from one amino acid to a keto acid. It's vital in amino acid metabolism and helps in the synthesis and breakdown of amino acids. Lastly, oxidative deamination involves removing an amino group from an amino acid with the help of oxidizing agents, releasing ammonia and forming a keto acid. This process is crucial in amino acid catabolism and energy production. All these properties, solubility, taste, light absorption, chemical modifications, and metabolic pathways, highlight the versatility and importance of amino acids in biology and chemistry. They're truly fundamental building blocks with diverse functions. Let's take a closer look at amino acids in solution. When amino acids are dissolved, their behavior is quite interesting because they can exist in multiple forms. The key point here is that amino acids have both amino groups and carboxyl groups, which can gain or lose protons depending on the pH of the environment. This means they can exist as positively charged, negatively charged, or zwitterionic molecules. Understanding their form in solution is essential because it influences how amino acids interact with each other and with other molecules, affecting protein structure and function. For example, at a neutral pH, amino acids tend to be in their zwitterionic form, where the amino group is protonated and the carboxyl group is deprotonated. This property of amino acids in solution ties directly into the overall theme of protein chemistry, as it helps explain how proteins fold and how they interact within the cellular environment. Recognizing these forms and behaviors is fundamental for understanding biochemical processes at the molecular level. Let's take a closer look at the isoelectric points of amino acids. This concept is crucial because it tells us the pH at which an amino acid carries no net electric charge. Understanding this helps us predict how amino acids behave in different environments, which is key in processes like protein purification. Each amino acid has a specific isoelectric point, determined by the pK values of its ionizable groups. For example, amino acids with acidic side chains tend to have lower isoelectric points, while those with basic side chains have higher ones. Recognizing these points allows us to understand how amino acids will interact in various pH conditions, connecting directly to how proteins fold and function. So, grasping the isoelectric points is fundamental in biochemistry and molecular biology, especially when studying protein structure and behavior. Let's take a closer look at the isoelectric points of amino acids. The isoelectric point, or pI, is the pH at which an amino acid carries no net electric charge. This is a crucial concept because it affects how amino acids behave in different environments, especially in proteins. Each amino acid has a unique pI based on its side chain and overall structure. For example, some amino acids have acidic side chains, which give them lower pI values, while others have basic side chains, resulting in higher pI values. Understanding these points helps us predict how amino acids will interact, dissolve, or precipitate at different pH levels. This knowledge ties into the broader theme of protein chemistry, as the charged state of amino acids influences protein folding, stability, and function. So, by knowing the isoelectric points, we can better understand protein behavior in biological systems. Now, let's take a closer look at peptide formation. When two amino acid molecules join together, they form a covalent bond called a peptide bond, which is a type of substituted amide linkage. This process creates a dipeptide. The key here is that forming this bond involves removing a molecule of water, a process known as dehydration, specifically from the carboxyl group of one amino acid and the amino group of the other. As we move forward, we see that 3 amino acids can connect via 2 peptide bonds to form a tripeptide. Similarly, amino acids can be linked sequentially to form longer chains, tetrapeptides, pentapeptides, and beyond. When only a few amino acids are linked, we refer to these as oligopeptides. But once we have many amino acids joined together, the structure is called a polypeptide. In fact, proteins can contain thousands of amino acid residues, making them incredibly complex and functional. Understanding peptide formation is fundamental because it explains how amino acids link to create the diverse proteins essential for life. Now let's talk about what we call an amino acid unit in a peptide. It's often referred to as a residue. This terminology comes from the fact that, during peptide formation, a hydrogen atom is lost from the amino group and a hydroxyl group from the carboxyl group of the amino acid. That's why we often see the term amino acyl, like alanyl, glutamyl, glycyl, and so on. In a peptide chain, the amino acid residue at one end, which has a free amino group, is called the amino terminal or N-terminal residue. Conversely, the residue at the other end, which has a free carboxyl group, is known as the carboxyl terminal or C-terminal residue. Understanding these ends is crucial because they define the directionality of the peptide chain, which is important when we talk about peptide sequencing and nomming. So, when we refer to polypeptides, the way we name and identify them often depends on these terminal residues, helping us understand their structure and function within the overall context of protein biology. Now, let's talk about the nomming of polypeptides. When we name these molecules, the key is to identify and name the amino acids that make up the chain. Typically, we use the standardized amino acid abbreviations, which are universally recognized in biochemistry. The sequence of amino acids is written from the N-terminus, which is the start of the chain, to the C-terminus, the end. This order reflects the synthesis direction and is crucial for understanding the structure and function of the protein. Remember, the nomming not only helps us communicate clearly, but also provides insight into the protein's properties and role in biological systems. So, in essence, accurate nomming connects to the bigger picture of understanding protein structure and function, which we've been exploring throughout this presentation. Let's take a closer look at the structure of a protein. Proteins are made up of smaller building blocks called amino acids, which are linked together in a specific sequence. This sequence determines the protein's unique structure and function. The basic structure starts with the amino acids forming a chain, known as the primary structure. Then, this chain folds into more complex shapes, creating secondary and tertiary structures. These folds are stabilized by various interactions, like hydrogen bonds and disulfide bridges. The overall three-dimensional shape of the protein, known as the quaternary structure, often involves multiple amino acid chains coming together. Understanding this hierarchical structure is crucial because it directly impacts how proteins perform their roles in the body. Whether it's enzymes catalyzing reactions or structural components in cells, their function is deeply tied to their structure. So, when we think about proteins, remember that their structure is both intricate and essential to their function. Let's wrap up with a quick note on the chemical nature of amino acids. Amino acids are quite interesting because they have both amino and carboxyl groups, making them amphoteric. This means they can act as acids or bases depending on the environment. Their side chains, or R groups, vary widely, giving each amino acid unique properties. Now, why are amino acids soluble in water but not in organic solvents? It's mainly because of their polar nature. The amino and carboxyl groups can form hydrogen bonds with water molecules, which makes them highly soluble. In contrast, organic solvents are generally nonpolar, so they don't interact well with these polar groups. This solubility difference is crucial for their biological functions, especially in the aqueous environments of our cells. So, understanding this helps us see how amino acids behave in different environments, tying back to their role in protein structure and function. Now, I have a couple of questions for you. Think about how this solubility impacts protein folding and stability.